us. Dr. Martino is a professor in neurology and co-lead of the Calgary Parkinson's Research Initiative at the Hotchkiss Brain Institute, University of Calgary. Dr. Martino is the secretary of the Canadian Movement Disorders Society, executive committee member of the Pan American section of the International Parkinson's and Movement Disorders Society, and co-editor-in-chief of Movement Disorders Clinical Practice. There was much more I could say, but I have put it in briefs. So at that point, I thank you, Davide, and hand it to you.
Thank you very much, Julian, and thank you for the opportunity to present. Can you hear me well? All good? Okay.
Absolutely fine. That's great.
Great. So yeah, this is an opportunity to talk about the link between the gut microbiota and its crosstalk with the brain in neurodevelopmental traits and neurodevelopmental disorders, which is by and large, it's not an easy topic, and it's got a lot of aspects that need to be taken into account. And in brief, this is what I'm going to be talking to you about. We're going to essentially say a few things about microbiome diversity during development. We're going to touch on what we think about neurodevelopment in its modern conception, and then how microbiome can influence neurodevelopment. And then we're going to get a little bit into the topic of neurodevelopmental traits and disorders from some basic mechanisms, then to understand how and why the microbiome is investigated in this context. And finally, what do we know about manipulating microbiota in neurodevelopmental traits and disorders? So a huge topic, which I'll try to summarize as much as I can. And I'm going to also apologize in advance. You will see a lot of very complex graphs, but don't be scared. I'm not going to, I didn't want to oversimplify them because I didn't want to give you the impression that this is a simple topic. It is a very complex topic, but I'm going to give you the take-home message from each slide. And so don't worry about it being overly complex, because I'm going to really summarize it for you as we move along. So first, a clarification about the terminology that we use, because it may not sound as immediately straightforward. So we're talking about microorganisms here, and these obviously include bacteria, fungi, viruses, and other less known, such as archaea and so on. All tiny organisms that can be found pretty much anywhere on this planet, from the glaciers of Antarctica to the deepest regions of the oceans, and also across our food system and in our body, obviously. And they're often just known for harm and dangers, like food poisoning or antimicrobial resistance or infectious diseases, obviously, of all sorts. But microorganisms have also many beneficial and positive effects for our health and well-being. They provide profound and essential services to the environment. And when we talk about microbiota, we mean an assembly of different microorganisms that live together in a habitat. And this habitat could be our body, our human gut, the human gut, or the soil, or any other environment or habitat. By microbiome, we mean a community of microorganisms that occupies a particular environment. And also, it's a term that considers how these microorganisms interact with each other in this environment, and also with their surrounding environment, which in our case are our own tissues and the different organs of our body, right, including the brain. So having said that, the first complex graph, but again, let me run it through you. So this is essentially a graph that is telling us one crucial thing, which is that our microbiome develops as we age and as we develop. So our microbiome, the one, and I'm going to say by microbiome, I intend primarily the gut microbiome, but obviously There are many microbiomes in many organs of our body, but I'm going to mostly focus on the gut microbiome because it's the one that we investigate the most and we think has important relationship also with our brain. So, as I said, the microbiome develops with age and it changes during development. And typically, one direction of change is its diversity. So at the very beginning of our life, it's very diverse, and as we mature through adulthood, it becomes less and less diverse, which means less and less different types of microbes live in it. Okay. Now, the microbiome, the microbial communities that develop in our early life as babies, really, um, they really are coming from our mothers, essentially. Um, so there is a lot of shared microbial strains between mothers and babies. It's a bit like genetics. So there is a shared material, not just genetic makeup, but also microbial makeup, and which suggests that there's a vertical transmission of microbiota from the maternal gut and the vaginal gut, the vaginal microbiota, or the breast milk to the babies. So as you can imagine, there are many different things that can influence how our microbiome our microbiome develops. First of all, well, there are also some genetic factors too. Our genetic makeup can influence microbiome. The gestational age, so the number of weeks when a baby is delivered, the mode of delivery, vaginal versus cesarean, makes a big difference. Early exposure to antibiotics and diet during pregnancy, they all have an influence on the developing microbiota of the infant. And there are some factors that can alter the development of the microbiome more than others. For example, antibiotics. So early exposure to antibiotics in early life can have a tremendous effect on how the microbiome develops. In particular, it can it's like antibiotics, they can obviously modify the diversity of the microbiome. They can wipe out a lot of species and so make it less diverse, which in other terms means an accelerated aging of our microbiome when we are exposed early to antibiotics during our life. So this is just to say that there's a nice gardening analogy. This is a gardening book. And I think it's a nice analogy with microbiome. There's no such thing as good and bad bacteria or fungi. It's not good and bad. It's just whether there's too much or too little of it. And things are out of balance. So the bad things, when things are out of balance, have an opportunity to prosper. The same principles of gardening apply to the microbiota environments. Now, again, another big graph, but this is only to say one thing, that our microbiome in the gut can communicate with the brain and can influence brain function. And so this happens in many different ways, but one important one is some metabolites, some small molecules that our gut microbiota produce. They can communicate and affect and exert functions in the brain. An example of these metabolites, which I'm going to mention several times during my presentation, are these short chain fatty acids. They're small molecules, they are little fats, but very, very tiny, and they can influence brain function significantly. Now, there are many different ways through which this can happen, as you can see, many pathways, but the most important one is the immune pathway. So, in other words, the gut microbiota communicates with the brain primarily through our immune system and through the activation or the inhibition of inflammatory mechanisms. So that is the key pathway. And then, of course, there are other ones which are less known or less clear, but by far the immune pathway is the most important. And through immune pathways, these metabolites of the microbiota can influence several behaviors and function, from motivation to mood, motor activity, learning and memory, food intake, and even the basic mechanisms of the brain development itself. Now, let's step away from microbiome for a moment, and let's talk about neurodevelopment and how we conceive it today. Now, neurodevelopment is a process of change of a number of brain functions, and you can see all these brain functions listed in this red oval on the right of the slide. So executive functions, regulating, learning, attention, emotions, impulses, sensory processing, motor execution, social behaviors, they all change over time with age, and this is what defines neurodevelopment. But what neurodevelopment really incorporates diversity, neurodiversity, simply because the development of these functions with age is different from one individual to another. So in the general population, it's different. So there's not such a thing as a non-neurodiverse neurodevelopment. It's always diverse. When, based on statistical terms, an individual reaches a maturation of a brain function in this area of the statistical distribution in the general population, this is when we call the individual with respect to that function neurotypical. Okay, this is also a term that reflects also societal norms, not just statistical terms, but everything that is non-neurotypical is called neurodivergent. Now, mind you, neurodivergent means that a person can have functional advantages, just as many disadvantages. So it's not a unidirectional link to functioning. It can bring advantages or disadvantages. And in a proportion of cases, this disadvantage can be impairing and cause pathology. But these are a smaller proportion. And this is when these extremes of neurodivergence may predispose to conditions like the more mainstream psychiatric disorders like depression or the neurodevelopmental disorders as such. But in general, here we are talking about neurodevelopmental traits, okay? Not disorders necessarily. Okay, now, neurodevelopment. When does it happen? Neurodevelopment happens from the moment in which we are conceived all the way throughout the first 2 decades of our life after birth. But you can see that a lot of stuff, the gray part here, is before birth. So there's a lot of milestones that happen before birth. And what is also important to our discussion today is that the immune system develops at the same time as the brain. Because I told you that the gut microbiota communicates with the brain primarily through the immune system. So it's very important that at birth, when the individual is ready to receive the gut microbiota, the individual has already undergone a significant amount of development both in the brain and in the immune system. And this includes the colonization of the brain by the so-called ambassadors of the immune system. We have ambassadors of the immune system inside the brain, and these are called microglia. Okay, these are immune cells that live in the brain, and they are very important in neurodevelopment, and they colonize the brain before birth. So this is just to give you the timeline of these processes. And the important thing is that the gut microbiome and the gut microbiota, better, play an essential role in various processes of brain development. And I'm going to give you some examples of the key phases and processes of neurodevelopment that can be influenced by the microbiome. Okay, let me clarify one thing here. All the processes I'm talking about that you can see summarized in this slide are processes that do not depend only on one factor. They depend on our genetic makeup. First and foremost, but then also they may be influenced by environmental factors, including the microbiome. But there's always a multitude of factors at play here, not just one. And this is very important because, as you may have heard, how more recently we are exposed more and more to discussions and debates about the environment playing a huge part into a number of neurodevelopmental traits development, particularly autism, right? As if the environment is the cause of autism, right? We're not saying this. We're saying that environmental factors can influence, can have a part, but of course we cannot underestimate the relevance of genetic makeup and of our own genetics to define these processes. So just some general example, just for you to understand the terminology that we use here within neurodevelopment. One is neurogenesis. Okay. So neurogenesis simply means the development of the brain cells, and in particular neurons, the ones that do all the work of our brain, basically. They have to differentiate from progenitor cells, from stem cells, into functional neurons. And this process of neurogenesis, which begins before birth but continues after birth, is critical for learning, memory, cognition, stress responses, and so on. Interestingly, there's even the possibility that some metabolites produced in the microbiota of the mother could even cross the placenta and act on the fetus during this neurogenesis. But this is only happening in a very small proportion. Most of the microbiota influence on neurogenesis happens after birth. Okay. Okay. And so typically one way we have to study and understand how microbiome influences neurodevelopment is through animal models. And the animal models that are particularly helpful with this are germ-free animal models. So in other words, rodents studied in the lab, they are made germ-free, so they are basically they're given birth in a complete absence of exposure to any kind of bacteria or microorganisms. And then we see how they develop. And then we can add microbial exposure to see how their development changes. This is the experimental context, a setting in which we learn about how the microbiome can influence neurodevelopment. And this is just to say that for example, some important molecules for our brain can be produced can be released by gut microbiota. For example, serotonin. You may be surprised, but serotonin, which is so important for our mood and emotion regulation, can be produced by gut microbiota. And in germ-free animals, on the other hand, when there's no microbial exposure at all and they live completely germ-free, what happens is that neurogenesis is a bit dysfunctional, and this process of neurogenesis can be altered. And in a similar way, antibiotics can also decrease or slow down this neurogenesis because they wipe out the balance of microbiota. Another process in the neurodevelopment that's influenced by microbiome is myelination. So our brain, when we are born, does not have myelin, which means that the connections between nerve cells that lead to the synapses and the transferring of messages or signals from one cell to another happens very much more slowly. And then after birth, our brain produces myelin, which is this sheath of material that enfolds the axons and allows signals to be transferred quicker, right? Now, if we don't have a correct myelination, we can have long-lasting neurological problems, okay? And And interestingly, the gut microbiota can regulate myelination too, to some extent, because they can control the production of myelin by the main cells in the brain that produce myelin, which are these guys here called oligodendrocytes that live close to neurons. So this is another example, another key step of neurodevelopment that can be influenced by gut microbiota. Another one is microglia. I mentioned it earlier, right? Microglia are the ambassadors of the immune system inside the brain. And they are very, very important because once they enter in the brain, they are responsible for the shaping of the synaptic networks, the connections between neurons. When during development, a gazillion of synaptic contacts is created, but some have to be eliminated because they are inefficient and they would only generate noise in our brain. And this is what's called synaptic pruning. It's literally like a gardening. We need to prune some contacts that we're not going to be using. And microglia does exactly this. It regulates the formation of synaptic connections and promotes the synaptic pruning. Microglia can be deficient in the absence of a balanced gut microbiome. So that's another aspect of neurodevelopment that is influenced by gut microbiota. And another one is the development of blood-brain barrier permeability. So blood-brain barrier is very crucial to separate and protect the brain from an excessive influx of all sorts of things from the bloodstream, and filters the exchange of molecules and nutrients between the brain and our bloodstream. If there isn't microbiota, then what happens is that this barrier is overly leaky, it's too permeable, and this can cause some brain dysfunction. But if we give metabolites that can be produced by microbiota, like these short-chain fatty acids that I mentioned earlier, this excessive permeability can be reduced and restored, okay? And finally, just to say, another function of the brain very important that can be influenced by microbiota is the development of the axis that controls stress response. That's another process of our development in which the microbiota can exert a regulatory effect. But overall, I just wanted to, again, reiterate the concept that just like neural development, also neurodivergent traits are multifactorial. They are sort of like the unique product of an interaction between our genetic makeup and different ways in which the environment we live in shape how we function. And so this is very important to keep in mind. And part of the neurodevelopmental traits that we see in neurodevelopmental disorders like tics, compulsive behaviors or obsessive compulsive behavior, hyperactivity, impulsivity, other types of autistic traits or behavioral traits that we see in autism, they all originate from the way in which specific networks in our brain are developed and mature. And the interesting part of all this is that the neurodivergent traits that we are interested in, they essentially depend on the development of pretty much the same set of circuits that interlink the cortex of our brain with a very deep and evolutionarily ancient part of our brain, which resides deep in our brain, called the basal ganglia. As you can see, these networks here, they look very similar because they connect roughly the same structures, but they're not identical. They are parallel loops that can overlap to some extent. But one, for example, controls tic generation. Another one controls hyperactivity. This one, called the limbic circuit, controls impulsive actions and compulsive actions. So this is all to say that all the neurodivergent traits and neurodevelopmental disorders that are close to us, our interest, are actually stemming from trajectories, in some way, non-typical trajectories of maturations of the same circuits in our brain. So neurodevelopment is very, very important to tics, to hyperactivity, impulsivity, OCD, obsessive compulsive behaviors, and autistic traits. And as a result, for what we've said so far, they can be to some extent influenced also by gut microbiota. And some of the mechanisms that I mentioned earlier are known in Tourette or OCD better than others. So for example, microglia. Why do I keep talking about these immune cells living in the brain? Because they are really very important, and they are typically overactive in people who have tics or obsessive compulsive features, from what we know, which is not a lot, but there are studies done in patients that have shown that there is a correlation between the distress that people with a diagnosis of OCD can have from compulsions and how overactive microglia is in their brain. And this can be measured because we have tracers that can directly link and identify overactive microglia in the brain using PET, which is one of those fancy nuclear imaging scans that we can do with we can apply in mostly for research, although there are also clinical PETs, as you know. But this PET I'm talking about is a research one. And there is also evidence coming from brains of diseased individuals, so post-mortem individuals with Tourette syndrome, that have shown that the most upregulated genes in the brain, in the basal ganglia of the brains of people with Tourette syndrome, are those that control the function of microglia. So there's a very big role of microglial overactivation in the genesis, in the development of neurodevelopmental disorders like Tourette or OCD. So why do we do microbiome research applied to neurodevelopmental traits? Well, essentially because we want to clarify how microbiota communicate with our brain to influence neurodevelopmental traits and to maybe predispose to the development of neurodevelopmental disorders. We want to understand the basic mechanisms, the cellular and molecular steps that explain this communication. We also want to clarify if there are environmental factors that interfere with this crosstalk between microbiota and brain, like diet or medications, or even the genetic makeup or social stress, other types of environmental stressors, and so on. Finally, obviously, we want to discover if we manipulate microbiota, can we do anything good to the health and the well-being of people living with neurodivergent traits? And which of these strategies are the most rewarding? These are the objectives of microbiome research applied to neurodevelopmental medicine. Now, let me tell you something a little bit about what's changed in the way we do this, right? So very few things in medicine has progressed, has advanced so quickly, like our research techniques to study and to decipher microbiome. In earlier stages, all we could do, we took, obviously, if we want to study the gut microbiota, obviously we studied stool samples, right? Fecal matter samples primarily, right? And so all we were doing at that time was to study these samples, and all we could do was capture changes between different groups of people with a condition, people without a condition. And we could compare the abundance of families of microbes or genera of microbes, so big groups of microbes, right? Now, thanks to the progress in DNA sequencing and in bioinformatics, we actually can resolve the identity of the species that can differ between different population groups and even get to the strains of these species and also their gene expression, which means how they function, how they interact between with each other and or with the host, with the tissues of our body. So in other words, the field has changed in a way like in a similar way as what we see when we are basically landing with a plane. At the beginning, we can only see just the entire towns or the cities or the villages from the distance, and that's all we can decipher. And as progress has occurred, it's like moving further down, and now we can start appreciating specific large agglomerates of buildings, then individual buildings, and then we're able to see cars moving on the roads. So essentially like a landing. And now we are at this level of we can discern much more deeply what's going on with the microbes in a given biological specimen. And again, the way we want to do this also because we are dealing with a complex spectrum. So neurodevelopmental disorders are a complex spectrum. They overlap. You all know that, particularly when we are talking about, unfortunately, pathologic presentations of these neurodivergent traits, these traits overlap significantly. There's a greater chance that a person who has tics, for example, may also have obsessive compulsive symptoms or may have some ADHD characteristics or even some autistic traits. And this is true if you take each and every one of these four main neurodevelopmental disorders, you know that there is a greater chance that having one of these four may lead to having greater chance of having another one, right? So there is overlap. And it's also possible that the same individual may have a pathologic form of some traits. So for example, having obsessive compulsive symptoms that reach the level of a disorder, and then have some hyperactivity or some tics, but that are not so severe to reach a degree of severity that we would call a disorder, right? So our approach to study microbiome research, to study microbiome in these conditions, is to pool all these traits together and study a large population of people who have at least one of these four neurodevelopmental disorders, so ADHD, autism, OCD, or Tourette, or tic disorders, and then take their unaffected siblings as control participants. And this is how we are proceeding right now. And we have actually reached one of the largest population samples that has this characteristic in so far that's been collected so far in a cross disorder fashion. And in each family, we basically collect information about many different things, about their medical history, about their gastrointestinal symptom history, and about how relevant each of these four major traits is present in all participants. And then we want to correlate this to what we find from different microbiome markers investigated by studying stool samples, basically. So we have completed this data collection at the moment, and now we're starting this huge analysis, which takes months. And hopefully, by the end of this year, we will start having some signals that essentially would teach us something about microbiome fingerprints of each of these four key neurodivergent traits: tics, autistic symptoms, obsessive compulsive symptoms, inattention and hyperactivity and impulsivity. And so we know there is obviously, this has been studied already quite a bit, not exactly in the way we are doing, but we know that there is evidence in the literature that shows that the composition of the microbiota of the gut in people with autism or ADHD differs from that of people coming from the general population, so controls, so to speak. And typically the change is always, there's some consistency in what the different studies, even though they're very different one from the other, are showing. So the consistent findings are that people who have these neurodevelopmental disorders have a greater abundance of bacteria that can promote inflammation and can promote inflammatory pathways in the body and can promote overactivity of the microglia. So they're all going in the same direction. So there's a greater abundance of these bacteria, and there is also a lower abundance of bacteria that have an anti-inflammatory property that could protect against excessive inflammation. And also, short-chain fatty acids that I mentioned earlier, that are such important metabolites for the crosstalk between the gut microbiota and the brain. Typically, in these neurodevelopmental traits, what we see is a lower number and function of bacteria that can produce these short-chain fatty acids. So these short-chain fatty acids are protective, are anti-inflammatory, and in association with these neurodivergent traits, they are less expressed by the gut microbiota. This is probably so far the largest study that was conducted in Tourette syndrome with microbiome, which is still, I think, less accurate than what we are doing. And it's also a relatively small study, less than 50 patients versus a similar amount of controls, not siblings, just separate controls, which differ from patients in many, many different ways. And so the differences are difficult to interpret because they may also depend on major differences in diet, genetic makeup. This is why we prefer to use to investigate enrolling unaffected siblings of the patients, because they are better matched for environment, right? And what this study, in any case, despite these limitations, what this study showed was that some pro-inflammatory bacteria were enriched in people with tic disorders compared to kids without tic disorders. But they also found that other bacteria could be increased simply by the exposure to dopamine receptor antagonists, the antipsychotics that we use to treat tics, like, I don't know, risperidone, aripiprazole, or others, right? Unfortunately, medications can have an effect on the gut microbiota too. And so we have to also keep that into account when we do these analyses. And this initial study has shown that, again, also for people with tic disorders, there is an increased abundance of those bacteria that promote inflammatory mechanisms, promote inflammation and overactivity of microglia, in a way that is quite similar to what happens in autism, in OCD, or in ADHD. And there is some initial evidence going again in the same direction, also investigating the gut microbiome of OCD and patients compared to a control population. Again, some bacteria go up, other bacteria go down. But the ones that go up tend to be more pro-inflammatory, and the ones that go down tend to be more anti-inflammatory. So this continues to be a consistent finding, even though we are talking about small studies that none of which can be considered the conclusive one, but all these signals seem to go in the same direction. So this brings me to the very final part of my presentation, which is, okay, There are some differences in the identity and the function of these microbiota between neurodevelopmental disorders and control populations. These differences may potentially affect crucial mechanisms in neurodevelopment. What can we do about it? Well, the answer is that we still don't know exactly what we can do about it and how intensely we should intervene on manipulating the microbiota in these populations. And we have to first of all start by just reminding ourselves of what are the strategies that are available. I mean, as you know, there are many strategies. There are many ways in which we can manipulate the microbiota. Unfortunately or fortunately, the microbiota are very easy to influence. Okay, very many things can do this. Of course, the traditional way to manipulate microbiota is, you've heard it probably, reading or across different sources, is the so-called fecal matter transplant. So you have donors from the population who do not have any neurodevelopmental condition, and they donate fecal matter because their fecal matter contains a bacterial environment, a bacterial ecosystem that is in some way maybe protective against neurodevelopmental because they haven't developed it. And then they are transferred to patients. Of course, all this first is done in animal models and in the lab, and then if it works in the lab, then it's brought to the clinical population. Now, so far, results on fecal matter transplants with in autism, for example, have been pretty diverse. So not really consistent effect has been shown. And if you can imagine, it's kind of non-specific and dirty strategy in a way, because you just take a whole environment and you are transferring it from one organism to another organism, from one body to another. We don't really know what we're doing by doing this. There are other ways which are more specific in some way. For example, we could the use of probiotics. Probiotics are essentially a limited number of species of bacteria that we know have some beneficial function, and we give them to an individual. We administer it, hoping that every change that these probiotics can produce in the gut microenvironment is going to be beneficial, which is okay as an approach. But the problem of this approach is that we don't know if that particular probiotic is really what is necessary in individuals with that condition. We have prebiotics, which means are essentially dietary factors that can, in a way, steer the microbiome in one direction or the other by promoting the growth of specific microbes and reducing that of other microbes. This is what we're doing with prebiotics. And if we pull them together, pre and probiotics, this is when we have synbiotics, which is another term that is used for pre-probiotic mixes. And finally, antibiotics. But as you can imagine, at the moment, antibiotics wipe out a large number of microbes. So we can't really use antibiotics to, it's not simply to wipe out an ecosystem, right? We need, and hopefully with machine learning and AI, we are now in the position to develop antibiotics that are so specific that they can target a single microbe, a single bacteria. This is probably the antibiotics of the future, and there are scientists, including in Canada, who are working on this particular type of designing new antibiotics. So the studies that have been done in autism with microbiota manipulation strategies are still relatively limited and heterogeneous. And they essentially aim to show if by giving pre or probiotic supplementation, we can modify behavior or gastrointestinal function in autism. As you know, people with autism may have quite a bit of gastrointestinal symptoms as well. They can have irritable bowel syndrome, can have functional constipation, and it's very, very common in autism. And so there are, as you can see here, just a number of studies that have shown that using, for example, probiotics that include this lactobacillus type of bacteria, some of these studies have shown that there's a possibility that this probiotics can enhance overall cognitive level in people with autism. Even some prebiotics that include particular sugars that belong to bacteria that can be particularly used for nutrition by some bacteria in the gut microbiota. These prebiotics might improve some behavioral symptoms. But again, the changes that these studies have observed are quite complex, are not always consistent, and overall, I think the take-home message is the last line of the slide, that large controlled trials using different types of measures, subjective and objective, are needed to show efficacy. In a similar situation, we can find in ADHD. There are studies that have used Symbiotic, this Symbiotic 2000. In Sweden, a Swedish study has shown that this symbiotic can reduce the comorbid autistic traits and the emotional dysregulation in people with ADHD. And it does this by increasing short chain fatty acid production. Another study in Israel showed that a multi-strain probiotic supplement given for 3 months was better than placebo in reducing hyperactivity, improving GI symptoms, enhancing academic performance in younger patients even. Another study from Taiwan showed that this probiotic that used bifidobacterium can improve some working memory, the performance on some working memory tasks. And finally, a recent study led by a group in Portland at the HOSU, the MADI study that looked at micronutrients, which are essentially prebiotics, in youth with ADHD, could lead to changes in microbial diversity and increase in the production of short-chain fatty acids like butyrate with micronutrients compared to placebo. And even in Tourette, this is the only study that has tried to demonstrate some effect of, in this case, again, probiotic, one specific bacterium, this type of lactobacillus, this very long name, Limosil lactobacillus reuteri. This is a study conducted in China, and they compared the effect of giving this probiotic to giving clonidine, which is an alpha agonist that has been used a lot in Tourette syndrome. And what they found was that this probiotic was more effective in improving tic symptoms compared to clonidine when added to the existing treatment of children with a chronic Tourette. These are all small studies. They all need to be confirmed. They need to be replicated in a larger fashion. And I would also like to understand particularly what this particular probiotic does, because in most cases, the choice of studying this type of probiotic versus another probiotic have nothing to do with the condition, are essentially based on very broad theories or hypotheses that some probiotics could have a beneficial effect in busting down excessive inflammatory activities in our body, and in some way, by doing so, help brain development. But it's all very, very non-specific and empirical. And this is my last slide, which essentially wants to give you a little, in a way, a little peek at the future of where we are aiming to leverage, how we are aiming to leverage the new information about learning about microbial fingerprints of neurodevelopmental disorders. So in my mind, if our study were to show, for example, that a group of bacteria in the gut of people with OCD is particularly defective, so it's less abundant, and we know that this lower abundance is associated with a reduction in a specific function that could be beneficial for the brain development. Now, then the next step would be to compound in capsules that we can call super probiotics the bacteria that are missing, that are less abundant, and try to restore their presence in the gut microenvironment. So this is a data-informed, data-driven selection of how to manipulate the microbiota in individuals that have a specific neurodevelopmental trait. In the case of this example, OCD, right? So this is, in a way, it's personalized or trait-specific manipulation of microbiota, which is more specific than just, throwing some probiotic to a group of people, hoping that it might overall help. And the technology is moving forward even more. I mentioned the super antibiotics earlier. So antibiotics that could be so precise to only wipe out one particular detrimental bacterium. At the moment, this is not almost impossible to do, but in the future it will be possible. And finally, bioengineering is doing incredible things by engineering microbes. So basically using DNA technology to change microbes and turning them into microbes that can perform actions that at baseline they wouldn't be able to perform. So using them almost like vectors of medications, almost like medications in a way, designing them using DNA engineering in such a way that we can twist and turn their function from neutral or even detrimental into beneficial. And this is really what people are dreaming of doing, mostly in cancer medicine. All the greatest advances in biomedicine start from cancer, always. So that's the field in biomedicine that is way ahead every other field. And then and the neurodevelopmental field has to follow the technological advances that are currently being already tested in oncology. But I think that this kind of synthetic biology is really the medicine of the future and the way. So these are the things that we really want to look after when we want to learn about new advances on microbiome science, rather than just hearing the latest study of a single probiotic that has been chosen almost randomly and may or may not have had an effect in a small study of a condition that we are interested in, right? So this was my 2-3 cents about the field, and I hope it was not overly it was not too overwhelming, and I hope it was somewhat informative. Thank you.
Thank you, Dr. Martino. That was absolutely fascinating. I'm sure